Let's start lesson 18, muscular organization. There are five important properties of muscle cells. Muscles have the ability to come together, right? If you look at those proteins, they have this contractility, which means shortening. But more accurately, the word is increase in tension. Contractility doesn't necessarily mean that muscle will shorten, but definitely the tension will. Muscles are responsive to stimuli. This is called excitability. Stimuli may be chemical, mechanical, or electrical signals. And muscles respond to generating electrical changes across plasma membrane. Conductivity is the ability of electrical changes to move or propagate along muscle membrane, plasma membrane, and it is like the copper wires in electrical impulses. Then the extensibility. Muscle may stretch up to 3 times resting length without damage. And the last one is elasticity. They have the ability to return to the original shape after being stretched. Now, let's take a look at the structure of a muscle cell. Most of the muscles have the same organelles as any cells with some difference. We want to start with this diagram. You see this myocyte. My means muscle, cyte is cell. So muscle cell. And the plasma membrane of a cell, in case of muscle, we call it sarcolemma. The cytoplasm is called sarcoplasm. And a smooth endoplasmic reticulum in muscle is called sarcoplasmic reticulum. It's a bit modified and it stores calcium. Right? So those are the words sarcolemma, sarcoplasm, sarcoplasmic reticulum. These are new to us. What about the myofibril? Please take a look at those myofibrils. Most of the cell of a muscle is uh, taken by these myofibrils, around 50 to 80%. And these are bundles of specialized proteins. Organelles are found packed between them. Sorry. Muscle cells differ from most cells. Let's take a look at this one skeletal muscle cell. If we compare it with the size of a red blood cell, it's much, much bigger, can be up to 30 centimeters. It's cylindrical, striated. You see those lines? And I will explain soon why are those lines there in skeletal muscle and cardiac muscle. Also, it is multinucleated. In one muscle cell, we get more than one nucleus. Fibers are composed of many cylindrical myofibrils surrounded by sarcolemma. And if I take one of those muscle fiber, this is what it looks like. Sarcolemma forms transverse tubule that enters deep into the muscle fiber. And transverse tubules are adjacent to sac of the sarcoplasmic reticulum. Together, they are called triad. You see in the right lower corner, there is a transverse tubule, and on both sides, those blue colored and sarcoplasmic reticulum. Together, they are called triad. And why is that important? The answer is coming soon. The set of transverse tubules and two terminal cisternae. The triad is important for muscle contraction because when we discuss how the active uh, those impulses going or generating moving through the muscle, that is where this thing will be discussed, the triad. Each myofilament consists of different proteins. Contractile protein to produce tension, regulatory protein to control when muscle fibers contract, and structural protein holds myofilaments in place and maintain stability of myofibrils. Look at those myofibrils inside the myocyte. There are three types of myofibrils: thick, thin, and those are the elastic filaments. Starting with the thick one, the red color on the right side, it is thick filament, also known as myosin. Many of those myosin filaments together, they will make this thick filament. In the thin filament, we get three things: actin, troponin, and tropomyosin. And then elastic fiber that is made up of titin. So once again, three types of myofilaments. The thick one is composed of protein myosin, which has two globular heads and two intertwining tails. You see in the lower part of the slide, two heads and one tail. In between, we have the neck, and there is a point called hinge. This is the part will be needed when we discuss how the muscle is contracting. Uh, that is vital. Myosin heads extend from tail on a neck, and neck is flexible where it meets tail at the hinge. For the thin filaments, three components: actin, troponin, tropomyosin. Those purple colored balls you see here, hundreds of them, these are actin subunits. And they have the active sites for the myosin head. You see that small hole within the ball? Keep that in mind. We'll need that. The active site for actin, and this is where the myosin heads will be attached. Some blue lines you find covering those active sites. These are tropomyosin. And two yellow things you see there, those are the troponins. Tropomyosins are rope-like regulatory protein, spiral around two actin strands. At rest, tropomyosin covers all active sites on actin. And the troponin, the second regulatory protein holding tropomyosin in place. Tropomyosin and troponin help switch on and off muscle contraction. We'll come to that point, how it works with the calcium. Elastic filament, number 3, composed of large structural protein called titin, which uncoils when stretched and recoils to its original shape. Assist in elasticity of muscle, runs through core of thick filament to stabilized myofibril. This is the elastic filament, and it is made up of titin. So the big picture of the skeletal muscle organization is like this. If I take the whole muscle, which is covered by epimysium, uh, that will be, but then part of that we take the fascicle out. Fascicle is covered by perimysium, and each single myocell, muscles, myofibril we take out, muscle fiber that is covered by endomysium. And inside that, we have myofibrils, thick filaments, thin and elastic. And at the end, you see how those actin and myosin are arranged here. Thick and thin filaments form alternating light and dark bands in a sarcomere. So, what is sarcomere? Sarcomere is the part of this filament from one Z disk to another Z disk. As you see in the upper part, zigzag line, that's Z disk. So, between two Z disks, we have one sarcomere. It is the functional unit of a muscle filament or muscle fiber. What else we have within that sarcomere? At the middle, we have this M line. So easy to remember, M is at middle. Part of the thick filament you see here, it belongs to H zone. The thin filament only area, that is the I band, and the zone of overlap with the H zone together, we call it A band. So we need to remember those different types of bands and zones. The I band contains only thin filaments. A band contains thick filament, the entire, along with the zone of overlap. H zone contains only the thick filament. So there's a mnemonic. You can try this to remember that I'm thin, he is thick, and together we are A band with M line at the middle, and we live in zone of sarcomere. So this is how to interpret this. I'm thin, that means I band, it includes only the thin part, thin filaments. Second part, he is thick, that means H band is for the thick filament, including, and then A band includes the whole thick filament, including the zone of overlap. M line will be at the middle, and from Z to Z, that's the area for sarcomere. So you can try this way or in your own way to understand those different types of bands and zones and lines. Now, after those things, let's go to the three-dimensional appearance of sarcomere. How does it look like? The sarcomere we know from Z to Z, this is how it looks like. And when those filaments, the thick and thin filaments, they slide each other, this is the moment the muscle will contract. This is called sliding filament mechanism of contraction. Take a look at this two hands, the fingers. In the first picture, the fingers are apart, but when they slide it past each other, the distance will be less. So similar way, the thick and thin filaments will interact and they will make the muscle to become shorter. During contraction, I band and H zone will narrow. As you see here, the upper part, when the muscle is relaxed, those things are very wide, the sarcomere. But when after contraction, they are coming close, so I band will be smaller, H zone will be smaller uh, than Z to Z. The disc, the sarcomere is also becoming shorter. The A band will remain unchanged. Myosin head grabs thin filaments and pull towards M line, that is middle line. Z disc move closer together and sarcomere shortens. So this will be explained in detail in next lectures. Last slide to know that sarcomeres arranged end to end within each myofibril. As individual sarcomeres simultaneously contract, the whole muscle fiber contracts. So this is all about chapter 18.